This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM This Week in Microbiology, episode 276, recorded on November 10th, 2022. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Do you have a hurricane ripping through you at this moment?
We have good old Nicole. She's come to visit, and she's dumping a whole bunch of rain,
All right,
which we desperately need.
All right. Well, you're in the hurricane belt, right?
Yep.
That's the way it goes. Also joining us from St. Louis, Petra Levin.
Hello. It's an unseasonal 75 degrees here, and there's students out in T-shirts and shorts.
Cool.
Or not. I can't decide. I'm a little unsettled about it.
Mm-hmm. It's so funny. So we have a library at Columbia called Low Library, and it has these huge steps going all the way down. You know, it's the famous one with the statue in front. And on a good day, everyone sits there, and it's called Low Beach. Well, so to our guest today, coming back for the nth time, no longer really a guest, but a frequent contributor from the University of Puget Sound, Mark Martin. Welcome back.
It's so good to be here with all of you with my quality quorum.
Quality quorum. Well, you have so many nice phrases. I really like that. Right? You have OMG.
Yes.
Overwhelming microbial greatness.
Yes. Quality
quorum. Anything else?
Yes, we do. WTM. Or what the microbe?
That one I haven't heard. And you make buttons out of these and give them to your students, right?
I do. I have buttons for both of those now.
This is a great way to engage students. I love it. Maybe I should do that. But I'm thinking Columbia students are too snobby, right? They don't want buttons.
You would be surprised.
And Petra's frowning. No, you don't think so?
I think everybody likes buttons. Actually, even now, I think they like better than buttons are stickers they can put on their laptops. Mm-hmm. That seems to be the thing.
Oh, I just got a bunch made for Microbe TV.
He's looking for the booth swag.
So yeah, there were a bunch of stickers. Where are they? Well, this is not Microbe, but these are Daniel's. These are Daniel's company, Parasites Without Borders. Right?
Yeah, that's actually water bottles and laptop stickers.
Yeah. So I had him make some Microbe TV stickers. I'm going to put one on my laptop. I always said I want a sticker on my laptop, but why not my own company?
That's right.
It's not mine anymore. Actually, it's a public company. It's a 501(c)(3). I have no more control over it, but nobody wants to control it, so that's good. Right? All right. So we are here to talk about two cool papers today, and we will have a little snippet from Michael.
So the title of today's paper is Over Half of Known Human Pathogenic Diseases Can Be Aggravated by Climate Change, and it appeared in Nature Climate Change in September of 2022, and it was authored by Maura, Mackenzie, Gaw, Dean, von Hammerstein, Knudsen, Setter, Smith, Webster, Potts, and Franklin. And they are located in Paradise, the University of Hawaii at Manoa, and a variety of departments from geography and the environment to marine sciences. As Vincent offered when we started this podcast, we are recording this the week the United Nations has convened a conference on climate change in Sharm el-Sheikh, which is in Egypt. It's going on right now, and I dropped the agenda for that conference into the show notes should you be interested. So I thought it only appropriate that we introduce you, our listeners, and the overall microbiology community of how climate change can affect human pathogenic diseases. It's also very appropriate that we just completed an election cycle where unfortunately many science deniers were elected to public office. And what many of them would offer is that while some may accept climate change, many will argue the full extent of the risk remains poorly quantified. And time is still on the planet and the human race's side to adjust and fix. Or should we take a page from the Romans and say it's later than we think?
Michael, they'll be the first to go to Mars.
Yeah, they will be. Well, today's paper and its companion, which is also in that same climate change journal, Climate Change Exasperates Almost 2/3 of Pathogenic Diseases Affecting Humans, are two of those must-read manuscripts from this year. The analysis paper, which we're principally going to walk through here this afternoon, conducted a comprehensive and systematic literature review where the fruits that the authors offer us reveal this startling fact that 58% of human pathogenic diseases are indeed aggravated by 10 common climatic hazards that are aggravated or sensitive to greenhouse gas emissions. Now, let's step back and unpack that sentence for a second. Over 58% of the pathogenic diseases. Now, we haven't had Elio on for a while to taunt us with a Talmudic question, but if there was ever a time where, you wonder about pathogens and climate change, here is one that I would have never guessed the number would be 58%. I would have ballparked at 30%. But in reality, this is not a new concept, as the authors point out to us. Potts and colleagues first reviewed this in 2005, where they described the impact of regional climate change on human health in an article, again, in Nature, where they reviewed on how there is indeed a connection between these climatic change inputs and health, albeit on a regional scale. The authors also offered us two other references, one from 2013 entitled Climate Change and Infectious Diseases, which was published in Science, where they offer us evidence and they're actually encouraging a predictive framework so that elected officials and people like you and I can actually make informed decisions about what we should be thinking about, doing research on, and encouraging our elected officials to do. And again, urgent mitigation is encouraged and early warning programs. Now imagine if we had an early warning program for something like a pandemic in 2013, or we listened to those folks in 2013. And, Vincent on TWiM has been talking about early warning programs for the next viral pandemic. And he's been talking about using our sewage for, of all things, looking for an old disease, polio, and asking a simple question, is polio running around the neighborhood? And lo and behold, it is indeed running around the neighborhood. And, but I think what really got me to encouraging my colleagues to talk about this paper is that the data from these two manuscripts truly slapped me in the face. And the data that are associated with Figure 3 from the analysis manuscript is entitled Climatic Hazards and Transmission Pathways for Diseases that are aggravated by these climatic change inputs. And by itself, it's a pretty spectacular illustration in which you don't really need all that much of a science background to actually digest. On the right, it's part word cloud. And the words, everyone understands how a word cloud works. The bigger the word, the more significant the hazard. And there's some pretty big words on that right-hand side. On the left, they got these 10 climatic inputs that are affecting the infections and infectious processes that they highlight in the middle. But the cool thing is the line thickness and the color gradients that the authors use are proportional to the number of unique pathogenic diseases and the proportional quantity of diseases, respectively. But what was truly impressive was that the author offered us, the audience, an interactive version where we, the reader, get to play mitigator, where we get to play with the inputs. So if you have an occasion to interact with a science denier, let them play with the levers from this website. Now, I'll drop the website into the show notes so you too can play.
This is a great graphic. Do you know that book, The Visual
Yes.
Display of Quantitative Information? This reminds me. It's so well done, right?
Oh, it's intuitive, and the users can navigate. And what this graphic is called is an interactive Sankey plot, and it displays how climatic hazards lead to pathogenic diseases via given transmission modes. And Mark, I'm sure, is teaching his undergraduates about how microbes move in the environment, whether it be a vector, whether it be droplet secretions. He's doing all that. And you get to play with what's actually going on. And the other cool thing is you can click on any disease named in the paper, and it'll take you to a case example, the citation, and then hold on to your pants a copy of the paper. So you don't even have to go on the scavenger hunt that you normally do with references. It's all there in this interactive site. And they have really done a service for society in doing this. And again, it's all for the purpose of transparency. The web tool and the background data are in the public domain because let's face it, folks, climate change affects the entire planet. It doesn't affect Charleston, South Carolina, though today it is. We're having a hurricane. And so now all of you know what it takes for data to slap me upside the head. And this paper certainly did that. I also like to offer that I'm also teaching an evidence-based practice course this semester to our dental students. And I've been co-teaching this with one of our librarians. And I showed her this paper after I picked it for this week's TWiM and offered to her, this is what big data really needs to do. And in the remaining time, I'm going to hopefully show you that this is not hype. This is not a cute little graphic, but it's strongly rooted in the fundamental science that we're all teaching our students. So what's the first thing these authors set out to do? And really, and Victor Rita is going to yell at me because I'm going to use this word. It's really a primer on how to write a grant. First, no study to date has attempted to comprehensively quantify the magnitude of human exposure to pathogenic diseases that can be influenced by climate change. So like we do when writing our grants, they attempted to fill this gap by performing a comprehensive literature review of diseases with a documented link, underscore the word documented, link to climate change. Again, what's the other important aspect of writing grants? Answering the why question. And here the authors offer an outstanding response, and I'm quoting, understanding the magnitude of the threat posed by the increased incidence of pathogenic diseases induced by climate change is an important piece in comprehending human vulnerability to climate change, which in turn will hopefully influence choice. And now, close quote, whether it be folks like you and I, or even the science deniers. They conducted a really straightforward systematic review of empirical observations of pathogenic diseases linked to the 10 climatic hazards by carrying out 3 complementary literature searches. Search 1 combined as keywords disease and each of the 10 hazards analyzed. The hazards span warming, drought, heat waves, wildfires, precipitations, floods, storms, sea level rise, ocean climate change, and land cover change. It sort of reads like the newspaper of the last 3 months. We've seen warming, we've seen droughts, we've seen wildfires, we've seen horrendous rains. I mean, this is really something to sit back and just reflect on. Search 2 combined each of the 10 hazards by each disease name listed in authoritative databases. They didn't go out and make anything up. They used Gideon, which is the Global Infectious Disease and Epidemiology Network database, and of course the CDC, the Center for Disease Control and Prevention, to help them identify what are infectious diseases. Search 3 was a data gap confirmation. And here they look for all combinations, disease names by climatic hazards, in which the first 2 searches failed to return any case examples, using alternative names of diseases, pathogens, and hazards. For this latest search, an approximation of the number of references scrutinized cannot be calculated because this latter search was variable in the number of papers looked at until the data they surfaced were up to about 200 citations reviewed. Overall, they found 3,213 empiric cases where climatic hazards were implicated in pathogenic diseases. That's a lot of stuff to read. All the empiric cases were related to 286 unique pathogenic diseases. They list all of them in the supplementary tables. This is open access, so you can go and plow into the data, of which 277 were aggravated by at least one climatic hazard. Although 63 diseases were diminished by some of the climatic hazards, 54 of them were at times also aggravated by other climatic hazards. So you just can't have one because the whole planet is changing and all of the inputs are impacting. There were only 9 pathogenic diseases that were exclusively diminished by climatic hazards. And this makes some sense. Warming, for instance, appears to have reduced somewhat the spread of viral diseases, probably related to unsuitable conditions for the virus, or because warmer temperatures lead to a stronger immune system in warmer conditions. For example, in the case of influenza, SARS, and COVID-19, in warmer climates, it may be better, as with rotaviral and noroviral enteritises. However, they also found that most diseases that were diminished by at least one hazard could at times be aggravated by another, sometimes even the same hazard. So they came up with some key takeaways. The first was climatic hazards bring pathogens closer to people. Now, it's no surprise to frequent listeners of any of the TWiM podcasts when you discover pathogens implicated. Shifts in geological range of species are one of the most common ecological indications of climate change. Warming, precipitation systems, for instance, were associated with the range of expansion of vectors such as mosquitoes, ticks, fleas, birds. I mean, after all, just look at how quickly West Nile spread across our country. And it sort of, was a stowaway on an airplane from Egypt, and voila, it's in New York. Several mammals were also implicated in outbreaks. We have viruses, bacteria, animals, protozoans. We have viruses like dengue and chikungunya, bacteria like the plague and Lyme, West Nile virus again, Zika virus, trypanosomes. We on TWiM have not really tapped into parasites, but I know over on TWiP they've covered trypanosomes and echinococcosis and malaria, just to name a few. And climate-driven expansions were also observed in aquatic systems, including cases of Vibrio species, for example, Vibrio cholera. And then this one is for Dixon, anisakiasis, which is a gastrointestinal zoonotic parasitic disease caused by the consumption of raw or undercooked seafood. And I'll drop into the show notes a reference to, you can read all the gory details of Vincent and Dixon having covered this on TWiP, but it's from ingesting nematodes from the genus Anisakiasis. Even though the reported cases indicate this is a rare disease, the true incidence of the disease could be potentially higher than what is reported in the literature, as often cases go undiagnosed as just a gastrointestinal upset. And then this final one is for Vincent and his TWiV colleagues, warming at higher latitudes allowed vectors and pathogens to survive the winters, aggravating outbreaks caused by several viruses, for example, Zika and dengue. And I saw that Vincent dropped into the show notes that he and his colleagues covered some of these climatic hazards on a recent TWiM episode, and that'll be in the show notes. That's TWiM 896, where they found how climatic changes led to movement of animals and microbes, which was leading to a greater number of diseases. So if you haven't checked out TWiM 876, I encourage you to do that. So another key takeaway, just like the first one, climatic hazards bring people closer to pathogens. Heat waves, for instance, we talked about it at the show opening, about the fact that it's warm in St. Louis and our students are out there gambling on the lawn in shorts and T-shirts. And so recreational water activities increase. And these have been associated with several rising cases of several waterborne diseases, such as Vibrio-associated infections, primary amoebic meningoencephalitis, and of course, good old gastroenteritis. Storms and floods and sea level rise cause human displacements implicated in cases of, disease we didn't talk about much, leptospirosis and cryptosporidiosis. We knew when the infrastructure collapsed in Milwaukee, we saw an uptick in crypto. And remember, crypto is really hard to kill by water filtration plants. So if it gets loose, our infrastructure, you don't want to be carting water around because crypto is not something that you want. And of course, there's a whole slew of other diseases that span the gap from Legionnaires that we have seen in places like Flint, Michigan, where due to changes in the water supply, albeit not to climate change, but to infrastructure failure, that we have seen it. And we're also witnessing that again in Jackson, Mississippi, where because of climatic changes, where we had those horrific rains this early fall, the water system was knocked out in Jackson, Mississippi due to the flooding. And of course, they had to truck water to the city of Jackson, the capital of Mississippi. And as we go, land use changes facilitate human encroachment into wild areas. And this creates new ecotones that brought people in closer proximity to the vectors and the pathogens, leading to numerous disease outbreaks, just like we saw in 2014 with Ebola. We're seeing scrub typhus outbreaks, as well as Queensland tick typhus, and of course, Lyme disease and malaria. The third takeaway is pathogens are strengthened by climatic hazard. Now, I'm not going to go into this in too much detail other than say, it's selection pressure, folks. That's what's driving this truck. It's as the climate change, we're selecting for microbes that can adapt to their circumstance. And the climatic hazards also enhance specific aspects of pathogens, including improved climate suitability for reproduction, acceleration of their life cycle, and all of these things go on. Ocean warming also accelerates the growth of harmful algal blooms. We've seen this in the Great Lakes with the toxal algal blooms associated with Lake Erie, the blue-green cyanobacteria, the dinoflagellates. All of this is a consequence of climate change. And the final two takeaways are people impaired by climatic hazards, and climatic hazards have also diminished the human capacity to cope with pathogens by altering their body condition. This adds principally due to stress. We've seen the stress in Jackson, Mississippi. I recently published a paper about PTSD and the stress induced by the water perturbations in Flint, Michigan, that was published in JAMA Open. And it's really quite remarkable on how something as simple as good water can influence someone's normal health. And the final thing I want to say is the diseases that were diminished by climatic hazards were quite few, only 9. And it really wasn't all that remarkable when you sat back and you thought about it. And the example that they offer, they commented that schistosomiasis infections were reduced by floods, limiting habitat suitability of the snail host, because schisto is transmitted by snails, and when you flood, the snail dies. But in other cases, floods increase human exposure and also broaden the dispersal of the host. And of course, droughts have also reduced the prevalence of malaria and chikungunya by reducing the breeding grounds. But in other circumstances, the mosquito density is actually even higher in these smaller reduced water pools. And so it's sort of, the model gets really squirrely when you begin to play with these things. All of this, folks, boils down to costs. And in their concluding paragraph, the authors point out an example that the cumulative financial costs of something as one pandemic, the COVID-19 pandemic, to the United States alone is going to cost us, the U.S. people, $16 trillion. I don't know about you, but if you bought a lottery ticket to win the billion, 16 trillion is 16,000 billion. That's a lot of money. That's a lot of money. So again, step back and unpack the statement that 58% of all infectious diseases known are being impacted by ongoing emissions of these greenhouse gases. So if you can drive one less trip, buy less gas, walk a little more, that's what we can all do so that the microbial world doesn't take us all over in the near future. So that's basically it, Vincent.
Michael, what are the naysayers going to say about this?
We have time. We have lots of time, and we'll make drugs. Big Pharma is solving all of these issues. But I don't think they realize, I mean, we covered the honeybee collapse on TWiM that was principally due to a bacterial infection. And we also have covered another incidents where there's mites in the honeybees. All of these things are, the planet is a living, breathing organism that takes multiple inputs. And this is really a good way to introduce undergraduates, I think, to the whole concept of science literacy and what it means to make informed, good choices as you're beginning your adult lives.
I was really struck by this because at first I thought it would be sort of selection on the actual microbes, but it actually is, it's the impact on the environment, their vectors, whether they're waterborne or not. And I hadn't thought about it in that way, maybe a little bit about the vectors, but the idea that the changing climate really impacts, I guess, their distribution or their dispersal is the big problem, it looks like, from this paper. And that was a really interesting way to think about it, because again, that's not a problem Big Pharma can solve at all. So that was I think a really important take-home message from this paper.
And I think we learned that lesson with COVID and how quickly COVID spread across the globe, because it was effectively an isolated instance of a focal point in China, and then boom, it was everywhere.
Right. I mean, I teach this class on infectious disease right now, and we just did one. Actually, one of my postdocs led it on talking about how we didn't understand how COVID was transmitted at the beginning. Like, is it droplet? Is it aerosols? This whole idea about standing 6 feet, but it's actually, these tiny things that doesn't actually make any difference, because these tiny droplets or really aerosols can travel hundreds of feet. And I think in this case, like, maybe it's sort of almost we should step back and think about transmission more than we think about actually killing the organisms. I mean, we need to do both, but the climate change contribution really emphasizes it's the transmission piece that we have kind of ignored at our peril.
And I heard about this paper. Vincent and I went to ID Week in Washington in mid-October, and it was in the keynote session in which they were talking about lessons learned from the pandemic. And they had a number of communicators talking about what we did right and what we did wrong. And one of the things we did wrong with the beginning of COVID is we should have said, masks work, but we don't have enough for the healthcare workers. So if you can stay home, stay home. But when we get enough masks, then you should wear them rather than what actually happened. And I really think we need to focus more on transmission and breaking the transmission cycle so that we don't need to resort to the nuclear weapons, namely the drugs.
Right. I mean, this paper, there's this couple famous papers from the late 1950s, early 1960s on TB even. And there was a big pushback on those papers, even though they show that it can be transmitted by aerosols using this complicated guinea pigs in sort of a center room, which is attached to the exhaust or the airflow from hospital patients with TB. Even though it's super clear, they still got pushback on whether it was aerosolized in the late 1950s. And it's just stepping back, all these things we take for granted. But then we don't have to go back past, March 2020 to this discussion of what now seems obvious. But at the time, and I think it's partly that the professionals don't want to disappoint us, but it's better to be honest than to disappoint us and have incredible unchecked spread.
I think that most scientists hate saying, I don't know. I have no trouble with those words. I think they're the most important words in science. And what I've noticed over and over again is that the young people that I teach are really attuned to that. And they're looking for, their BS meters, for lack of a better term, to kind of do their thing. So I would argue that being open with people is always better than hiding anything. At the same time, I've never been in charge of a lot of public health. Right? So it's very easy. It's very easy for me to sound critical, but at the same time, we should be cautious about that.
I think
I'm sorry, Petra, but the way that you described, just being honest about the masks, that's what I was telling everyone at the time.
No, exactly. I mean, in the masks, it's one thing to be like, in the end, we were, all strapped inside, but maybe it would have been better to just say, once we have masks, I don't know. It just seems like such a small thing and easy to do. And if we can do it, assuming we had enough masks, it would be better to just say, look, better safe than sorry. It's clearly respiratory. Masks work with other respiratory diseases. Just rip the Band-Aid off and tell people the truth, which isn't that hard to deal with. But yes, I agree. It's another thing to tell them, I'm sorry, I misunderstood how DNA replication worked, or this small aspect, right? I admit I'm wrong all the time, but it is much harder once a lot of people have gotten sick and hospitalized, which hopefully has never happened when I made a mistake about DNA replication in a lecture.
That's right.
Well, thank you, Michael.
Thanks, everyone.
Very good. And now, Mark, what do you have for us?
Well, the first thing I want to say is now I feel, very low about talking about fun stuff after that sobering and
you have to uplift this, Mark.
Yeah, but I did want to say before I begin that I'm going to be having my students play with that program, for lack of a better term, that app. I think they'll really enjoy it. So thanks for that.
You're welcome.
So I would like to say that here in Tacoma, it's nice and cloudy. It's 43 degrees Fahrenheit, 6 degrees C. And it's good to see everyone doing well. And I'm doing wonderful with less cryo freezer noise in the background. And I want to thank everyone for letting us start a few minutes early because I had to finish a lecture on bacterial photosynthesis, and I'm ready to shed some light on today's topic with WTM and OMG, as the saying goes. And I really like this paper, and I do worry that this will turn into a nerd fight, not here among this quality quorum, but in general. Now, this topic is from Mazzano and Company at the National Institute of Technology in Tokyo. And I want to give credit to Mizuno, Marie Nagamura, Koga, Hirayama, Fukuoka, Tanaka, and Morikawa, who've worked on this. And apparently this work has been going since 2008. It's published in eLife in 2022. And I consider the title of this to be completely and utterly microbially metal. Novel, anytime you start with novel, it's great. Novel multicellular prokaryote discovered next to an underground stream. Now, I just love this to pieces. I have many things to say about this paper, but I'd like to ask the people here, I'd like to ask each of you a question. How do you define multicellular?
I knew you were going to ask something hard.
Oh, this is no right or wrong answer. I just wonder how whoever wants to go first. I'd love to hear what you say.
Well, I grew up at Indiana University, where we had on faculty David White, who worked on Stigmatella aurantiaca, which is a multicellular creature. It's really individual bacteria that have behavior. Mm-hmm. You know, they eat other bacteria. You would feed them E. coli, and they're in the classification of the myxobacteria. And so is that really a multicellularity? Because they go through a developmental program. It was the beginning of the days of understanding different sigma factors and bacterial programming. This was the time of Rich Losick and all of his fundamental observations with sporulation. It was really an exciting time. And so that's really my bias in multicellular ism. It's really the same bacterium with just a little different shape, but they're all on top of one another. And the naysayers are going to say, how is this not just simply a homogeneous biofilm?
No, I agree with that completely. Thank you, Michael, for that, because this turns into such an interesting topic in general. Vincent, would you care to weigh in on this?
I would look at it very straightforward. Multi-cell, a lot of cells living together. Could be a biofilm, could be a filament, or it could be more complicated where the cells have different functions. I don't think multicellular is necessarily one or the other. That's all encompassed. That's how I would look at it.
So I love these scientific debates. It's interesting to watch people go back and forth. And I'm watching Petra laugh because I know Petra has strong opinions on this.
I don't know if I have strong opinions. I feel like this is all semantics to some extent, right? I mean, honestly, if I were going to tell you a bacteria that has a multicellular lifestyle, sometimes, I mean, I think most bacteria at some point find themselves in some kind of consortium of some kind, which even in the gut, right, the bacteria, one digests this and then its waste product is digested by the next one. I mean, they're dependent on one another, but you could have a definition that's multicellularity is where you have differentiation and division of tasks within the same microbial species. So like Myxococcus would be a great example of that, or Stigmatella. So I don't know. I don't have actually a strong opinion other than I think, there are many lifestyles for bacteria. That's what makes them so fantastic.
I could, first evolved, last extinct. That's the whole situation there. And I completely love your answers. And
what about you, Mark? What about you? What do you say?
Oh, I talk about this a lot in class with my poor students. And often they'll say, do you think it's this or that or this other thing? And I'll say yes.
You're mean. You're mean to them.
No, I am not. I love my micronauts there. And by the way, I'm waving at them. I don't know that they'll listen to this. My own family doesn't listen to podcasts that I'm on. So we'll see. But I think we're all aware of the trichome of cyanobacteria. And there's that differentiation into heterocyst where they wall things off. And you can use nanosims to find out that nutrients are being transferred. That's a wonderful thing. I think that Myxobacter shuffle during predation, where you have stems and spore-forming cells. I'm even in love with the mat-forming bacteria Pseudomonas fluorescens makes, that you develop cheaters in the mat that eventually sink the mat. The whole point is there's a group of things all interacting. And it's like the poem by John Donne that no man is an island. Well, no microbe is an island. I mean, we're all interactive. I sometimes worry about biofilms as being viewed as being multicellular, but I'm okay with it. As again, as Petra says, it comes to semantics. But what I often talk about with my students, not to get Margulian with you, is a kefir grain multicellular because you can't reassemble it. You can reassemble a sponge. You can separate them into cells and they come back together, but you can't reassemble a kefir grain. So I don't know if I go quite that far, but it's a really interesting nerd fight. And I love to see students, like when I used to argue about the definition, I would bring the debate of what a prokaryote is to my students. And, you know, the Norm Pace, Barney Whitman thing was so much fun to discuss with students. But this is a wonderful paper to begin with for this purpose, for this level of discussion. And in fact, I think that there was a letter that arrived on Microbe TV about asking for recommendations of papers that would be good for undergraduates. And I think this is. And I would argue that the eLife editorial illuminating a new path to multicellularity, I'm sure that irritated a bunch of people. It's a wonderful editorial. The science editorial has beautiful pictures. First off, I love the title. Odd cave bacterium forms a multicellular body like plants and animals. Now, I don't know about that, but pretty pictures. And on that same topic, when I was looking around, I don't know if you've all seen this article by Julia Schwartzman and Otto Cordero. Bacterial growth in multicellular aggregates leads to the emergence of complex life cycles. And it's Vibrio splendida in the ocean that forms what I could only call a snow globe with motile bacteria in the center as it breaks down alginate. And the reason I bring all this up is you have these multicellular assemblages doing things. And when do you call that a multicellular organism or just a bunch of neighbors? Sometimes freeloading neighbors, but neighbors nonetheless. So I'm going to put those references up on the shared drive so you can put them up. But I think that the eLife and Science editorials, they argue pretty strongly, and maybe the science went a bit much. They're very cautious to quote very famous scientists that aren't quite so convinced. And that's good. That's wonderful for a lot of reasons. But the eLife article does a masterful job in the introduction of laying things out. I cannot emphasize that enough. This isn't, we found this cool thing. They went on to talk about the background to things. It's like a master class of definition. And the references for this paper are outstanding. You've got Jim Shapiro, who thinks every colony is a multicellular organism. Dennis Clausen, who works on streptomyces. Nicole King, oh my goodness, right? Paul Rainey, the OG. Hazel Barton, who's forgotten more about cave microbiology than I'll ever know. Our friend Harmit Malik and Ben Kerr. So my point is, these people really worked hard on putting together a solid introduction and discussion. If you look at figure 1 in particular, look at the paths that they list there for what they're calling prokaryotic multicellularity, both aggregative and clonal. And one of the reasons that I think people don't take this subject so seriously is that if you look at Caulobacter, which I dearly love, you can say that that's relatively simple because of the stalked and swarming cells. You can look a little bit at the filamentation that you might see in cyanobacteria and say it's not quite the same. But when you start to get to patterned multicellularity, which is relevant to today, and also this business, and another time I would love to have someone dive deep into these magnetotactic prokaryotes that form, which look like little magnetic footballs. Really amazing stuff. So I think they did a really nice job setting this up. And then they went on to something that gets a little bit evolutionary. And I'm cautious when I say this. When folks debate the origin of multicellularity, we can talk about snowflake yeast, and we can talk about the mat forming organisms, all this kind of thing. Comes down to this phrase, ecological scaffolding. And what that means is that you have environmental factors that can lend extrinsic selective pressure to this evolving population. And that brings us to the paper, because what this is a cave organism that deals with multiple cycles of flooding. And this brings us back to environmental change in a way, Michael, right? Mm-hmm. So I have to start the story. I love the idea of caves, though I am claustrophobic. Caves are fast. The microbiology of caves is fascinating. Every time Hazel Barton gives a talk, I am just enthralled with the stuff that she finds. Such an adventurer. But the interesting thing is, and I think this is related, is that most caverns are somewhat oligotrophic. So there's a lot of competition for rare nutrients. And I know from discussions with Hazel, all kinds of strange things have been found. But Mizuno's group is interested in trying to do bioprospecting in unusual places, although sometimes that unusual place is in your backyard.
So the story starts with the workers here. We're talking again about Mizuno's group, bioprospecting in oligotrophic caves, and they're looking for new and interesting microbes that have useful properties, presumably for biotech. Hazel Barton, I hope you're listening to this. In any event, strain HS3 was found, and I love this expression, a dripping limestone cave wall in the Hirao Karst Plateau in Japan. Now, I do have to say parenthetically that when folks do bioprospecting, I used to do that a little bit in biotech. Sometimes it's not in an exotic location, but I'm delighted they found something in an exotic location. So in a nutshell, this strain that they found, HS3, appears to develop a distinctive multicellular structure due to environmental changes, and then disperse a different morphological form for that dispersal. It's thought to be due to recurrent flooding in the caves. And I think I got cut. I had an issue with recording when I was saying that I love wild microbes because they have all kinds of interesting adaptations that are often lost during cultivation. It's a gram-negative obligate aerobe with a single polar flagellum, and it's apparently related to Niceraceae, which is nice to know. And they named it, and I'm going to have trouble here, Geongupia saculi. And the saculi tells you a little bit about what's happening. Now, what's interesting when you look at the paper are some of the beautiful photographs. On agar media, and they're using in particular something called R2A medium, which is used primarily for waterborne microbes. I use it when I'm looking at the organisms that live in students' water bottles. So, water bottle buddies, what's wrong with that? I'll send you stickers, Vincent. In any event, on this agar media, you get these really beautiful colonies, transparent colonies, and they're iridescent. And what I found fascinating is that I have seen Cytophaga from seawater do very iridescent colonies. And this has to do with pigments that form a repeated structure, so they diffract light in a particular fashion. But I've never seen anything quite like this. The cells are oriented into what they call a nematic pattern, a liquid crystal arrangement. And what I thought was fascinating, and I hate to jump around, but this is important. They took a shoe of science and they beat the you-know-what out of this system. It is so amazing to me. Even though an editor calls this qualitative, and I understand why, I'm so impressed by the simple and complex levels of research that they applied to this. So in any event, many people would look at something like this, because I would have, and thought it had to do with the orientation of pigment molecules. For example, Serratia marcescens that makes the pigment prodigiosin often will get an iridescent sheen to it because of the way the pigment piles up in the cell wall. But this is actually the bacteria orienting. Mm-hmm. And they find that out. This is a genetically tractable organism, which just annoys me no end. I'm happy for them. But I have so many interesting microbes that will not yield to my wishes. And so what they did is they electroporated the darn thing with a broad host range plasmid carrying GFP under a strong promoter, and they're actually able to show that this nematic pattern, this liquid crystal arrangement, is simply due to the filamentous bacteria organizing. And then it just kind of sits there and grows, develops bumps. The pictures are outstanding, by the way. And then you start to get the loss of nematic structure. And then inside that nematic structure, you get a different shape. And they're calling them cocobacilli, which to me just simply sounds like short bacilli. Start to bulge out, and even the adjacent colonies seem to do it. What it looks like is that the filamentous form acts to support the cocobacilli as they're forming. And there's a beautiful, it's the third video in the supplementary information. When you take these, I don't know, they look kind of, I'm sorry to say it this way, they kind of look like pimples on top of the colony.
Nah, they look like edamame on top of chow mein noodles.
Well, that's different, but okay.
Well, pathologists always use food references to describe things.
Okay.
Funny. That's funny.
Well, in any event, when they submerge the plate in water, the cocobacilli are released in a stream.
Mm-hmm.
And they have video of it. And of course, they're thinking this is related to flooding in the caves. And the videos, I mean, they can show how the colonies grow. They can show the formation of the cocobacilli. And then they show that business of streaming and the videos. And figure 4 shows kind of a graphic view. It's a remarkable study and well worth everyone's time. I could easily see someone saying that this is no different than fill in the blank of some of the differentiating microbes we've already discussed. I mean, you've heard me say before that one of the things that I hammer into my students, long-suffering students as they are, is that there are the 3 deadly centrisms, and the third of them is coli centricity, that many microbiologists suffer from, the idea that we think that all microbes are like E. coli. Heck, I don't think E. coli is like E. coli in the lab, right? So
it's not when you get sick.
Yeah. And so for me, this is a step along the path to what we would call multicellularity. Again, from every level, from the references to the techniques that they're using, I was struck by the fact there's no external funding for this research, incidentally, which must have mean it was a labor of love. What do you folks think? Is this just another strategy, or is this on the path to multicellularity?
There's so much here to unpack. The releasing in water piece by itself is a lifetime of work.
Yeah.
Of trying to understand the selective pressure that evolved into a nozzle. I mean, that's effectively what they're implying. The whole issue, and some of the figures are just spectacular. I'm looking at figure 3, panel D, and it's just spectacular in terms of the color shifting.
Mm-hmm.
And since this is all in the public domain, you can get where I came up with the edamame references with the noodles and figure 2, panel E. And it's really a phenomenon of, if you just unpack it and ask, you ask 2 fundamental questions. How do you know and how do you show? And it requires knockouts, it requires mutants, it requires GFP vectors to ask fundamental questions. I mean, this is a great model system to really try to tease apart some of the fundamental questions that Mark is trying to get us to go down the garden path with about multicellularity.
This figure 3D, which I agree is really beautiful. It was actually, this figure was reminding me of the paper we did, the last TWiM.
Yeah. Yes.
About how the bacteria in the colonies actually sort themselves and give the colony shape. I think it's very reminiscent of that in a different way, but the same idea is that it's, not a outward process, like
Well, I
concentric
mean,
process. It's cool.
it's so interesting to me to see the patterns that if you ever worked with an undomesticated, freshly isolated bacteria, it's probably better to call it that, you'll get these interior patterns that you often see in colonies. Now, sometimes the structure of a colony is really more of an issue of physics than anything else. I know that some of the, quote, mushrooms that you see in biofilms are really more of a hydrodynamic kind of situation. So the question is how much of the patterning that we see is actually due to the programming. When you have these individual filamentous cells that actually kind of sidle up to each other and like are oriented, that's interesting. I do worry if they're going to lose all this, if they cultivate it more and more, number one. Number two, I don't mean to say, and I don't want Jim Shapiro, because James Shapiro really influenced me many years ago with the idea of pattern formation in colonies. But I am worried a little bit that colonies may not represent how this grows on a cave wall. That I don't know.
Mark, why do you say this is on the path to multicellularity? Looks pretty damn multicellular to me.
Well, I think that part of what people want to see is distinct compartments, and I mean that in terms of functionality.
Okay.
So I would agree with you. It looks multicellular, but someone could look at this and say, how does this specifically differ from Caulobacter, and maybe with like a whole garden of Caulobacter? But the thing that convinces me that we are on the path is the way that the original filamentous cells form as kind of a support. Again, you can look at that a little bit like a stalk for Myxobacters.
Yeah.
And then you get this different form that gets expelled. How would you study that in the wild? I don't know. I do know that when you grow it on plates and you flood them, you get those beautiful videos. So I almost want a microscope on the cave wall. And I want to see, not when it floods, that's not what I mean. But, I think it's a really interesting idea. I look at this in very much like some movies I've seen. I look at it and to my mind, it says this means something. And I think it does. So when you think about differentiation, and we have a little, and this is very similar. If you look at Anabina, you can find mutants of Anabina that have more frequent heterocysts and rarer heterocysts. So there clearly is something controlling pattern formation during the differentiative event. So that's why I hope they can continue to work on this. But it fascinated me because of the beauty of it and the clarity with which they described things that they did. So I can be convinced. I was just trying to err on the side of being a devil's advocate.
Yeah, they call it a unique multicellular behavior, right? That they do think it is.
Yeah.
That's fine. They say, example of an extant prokaryote with a unique multicellular behavior based on liquid crystal characteristics, timely regulated development, and potentially involved in recurrent dynamism in its cave environment.
Yeah.
I'm sure they'll study it, right?
I hope so. Petra, what were you going to say?
Oh, I always worry about using the word unique or novel when describing bacteria or microbes in general, because it's only previously unidentified by us. That's, I guess.
So you're right, Petra. When I was in graduate school all these years ago, the first paper that I was first author on, I thought it was so cool, and I kept saying interestingly. And Simon Silver was the editor, and boy, did he slap my hands.
Well, that word is also just awkward, I think, is also. I put it in papers, and then I always end up taking it out when I remember. But I mean, actually, my postdoc advisor would say it's not novel to the organism.
Yeah, that's true.
It is. This is really, I think, beautiful and unexpected, I think, is a better way. You never know. It's a surprise. And that's what I think is so great about discovering new bacteria and new ways that they live. We can't imagine it until we find it, and then it makes sense.
So one of the things that I talk about a lot, because I'm interested in variable phenotypes, is if you read the old literature, like Behring's old literature, he will find, say, luminous bacteria or pigmented ones, but they weren't stable. They would throw off sectors or wouldn't be reliable, and you would keep restreaking until they got something to be stable. And I understand why it was done that way. But I believe that variability is much more the rule. But in order to study it closely, we tend to lose it. So I would make so many frozen stocks of this ASAP.
Mm-hmm. So what are your thoughts on small colony variants?
Yes. My thoughts on that are, and you're talking about staph I agree. I've seen Micrococcus do that. So it's a relative. So I had to 16S it to make sure it was the same thing. And, I do think that growth in the lab is a strong selective pressure.
So let me turn this question around. Is a small colony variant on the path to multicellularity?
Within a tetrad, is that what you mean?
Well, because Staphylococci, the way they grow on top of one another and their irregular planar division, as Petra pointed out in the beginning, it's all an issue of semantics, I think. But it really trying to decipher small colony variants, because clinically they're remarkable. Small colony variants are a remarkable clinical phenotype that have consequences for antibiotic prescribing and other things.
Right.
And so you're absolutely correct. Growing what we do in lab to identify the pathogen versus what's actually going on in the patient is very, very different. And there were a lot of talks at ID Week on small colony variants. And, I hadn't thought about it until I began to read this paper and began to, think. And that's, I guess, what I most enjoy about doing this podcast is you force us to think differently by picking some of these papers.
Well, Michael, I want you are you familiar with, it's a bioinformatic program called Patrick?
No. So
what Patrick can be used for is if you have a genome sequence of a reference organism, it doesn't have a K on the end. So it's Patrick as if you're very fancy. And you have a reference organism for which you have a published genome, and then you have something that you've put through whatever. And you want to see, like, if it grows better at a higher temperature, or maybe it's more resistant to something, or it makes a mat. Regardless, you can get that genome, and Patrick fishes out the changes that have occurred. And you can do it for like $80, which is what I do with some of the stuff in my lab when I'm looking at variants in luminosity in some of these luminous bacteria. And you find these genes that are changed, and you have to ask the question, what has changed specifically? So with your small colony variants that you're mentioning, I wonder if people have done that.
Oh, they breed true. When you grow them on rich medium, they come back to normal.
Yeah. So that would be more a matter of transcriptomics to know what was expressed in that case. Certainly.
So, Mark, what would be, in your opinion, the next experiment they should do?
Oh, man. I would want to set up some kind of analog to what they're seeing on the limestone wall rather than a Petri dish. And I know what they would do. The fellow who's at Slava Epstein and Kim Lewis at Northeastern. I have that right, don't I? Oh, good. Everyone's nodding. So they developed years ago they were concerned about microbes that were unculturable, and they found this way of creating essentially a Petri dish that was in contact with the environment nutritionally. So I'm wondering if they could use that approach to look at these situations in the cave. And you might say, what are you going to do? Wait for it to flood? Well, you could imitate that. In other words, to see, does it act the same in a more natural environment as it does here? In addition, I'm curious about the genome. I'm curious, since it's so tractable, can you find, and I am just picking this out of the air, I find that type 4 pili are often involved in group behaviors. I would look for type 4 pili and just do a knockout to see or a knockdown to see if it would have an effect, especially because you can look at some of the behavior on a plate. That would be very interesting to me. I don't know about a standard, like, I can't imagine a gazillion plates with this looking for mutants, but I do think a targeted approach would be interesting. Certainly they're modal, so the role of motility, both flagellar and non-flagellar, could be easily looked at. But, this is like the joke to a carpenter. I'm kind of an old geneticist. So like a carpenter solves every problem with hammer and nails. So I do that with transposons and sequencing.
Right, which has gotten much cheaper.
Yes.
And much faster.
But I love how this looks. I love the liquid crystal approach to it. I want to see if it grows on limestone this way.
Okay. Thank you, Mark.
Most welcome.
Love it. Really cool paper. Thanks for suggesting it. That'll do it for TWiM 276. You can find show notes, microbe.tv/TWiM. Questions, comments, TWiM at microbe.tv. If you like what we do, consider supporting us, microbe.tv/contribute. Michael Schmitz at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone. Enjoyed it.
Petra Levins at Washington University in St. Louis. Thanks, Petra.
Thank you.
Mark Martins at University of Puget Sound, which is in Tacoma, Washington. Thank you, Mark. Good to have you again.
You're very welcome. I'm always appreciative to be TWiM adjacent.
TWiM adjacent. And you sound good today because we sent you new equipment so you can keep it for next time.
Thank you so much.
My pleasure. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.